Hello, everyone. Welcome back. Today morning, we have done some discussion about the structure of flower, and we also saw how the male part, that is the anther, and we saw the transverse section of it, and also we had seen how the gametophyte in it develops. So this is the second part, and in this video, we will be covering some very easy, interesting, and utmost important topics, which you can definitely expect some objective questions for your NEET, and also some important concepts from the board exam point of view. So, in this video, you will be studying pollination, cross pollination, what is the difference between it, how a flower can adapt to it, or how it can prevent it in some cases, followed by two very important concepts, that is pollen pistil interaction and double fertilization. I hope everything is audible and clear. So let's start. I'm Janani Saptharishi and I'm going to be teaching class 12 biology, and I'm associated with Unacademy. So what will Unacademy give you? So these are the benefits for subscribing into Unacademy. Okay, you will have your live classes daily. Okay, you'll have n number of live classes. You can join in any time you wish. All right, you'll have get access to live tests, quizzes, and question banks, all set by the top educators. You will have structured courses. All right, so by that I mean, like, if you subscribe through my code, that doesn't mean that you will study only under me and you will study only biology. You can prepare for any competitive exam. All right, you will have unlimited access to all the content. Like, if you're preparing for NEET, you'll have unlimited access to physics, chemistry, biology, and all the subjects you want. Everyday live classes with the top educators on board. So even so, if you enroll, it is not that you have to study only under me. With your ease, your comfort, your choice, you can choose your educator with whom you are comfortable and keep your learning on the go. We also offer you a very comprehensive syllabus, and along with that, the Unacademy Learning app, what you all you have to do is go to the Play Store, download this app, and get the subscription. So this is the app you need to download, and please get the subscription of the same. Then you'll go to the Telegram and copy the following link. This link is given in the description box below. So another interesting thing I want to tell you, that if you use the code Janani, that is my name, you can avail and 10% off on the course. That is for NEET biology. So you will get a 10% off on all this. Okay, so these are the different fee structures for the month. The most economical one is the one of the 12 month. All right, it is just 15,000 rupees for entire 12 months, plus you will get an additional 10% discount. So for an examination like NEET, I highly recommend you to take this one because you need time, especially if you're in class 11. You can prepare and very thoroughly, and it will be really helpful. If you're in 12, you can choose any of the crash courses. So let's begin. So today's topic, what we'll begin with, is pollination. So we have learned pollination definitely in lower classes. We know what it is, and now we need to know how it happens. Why is it so important? What are the different types? How it is carried out? What is the difference? All right. So these all we will be covering. Hello, Mr. Utkarsh. How are you? So, pollination basically happens in all flowers, okay, in all fruiting plants. Pollination is necessary for the flowers to go into produce fruits, and you plant any plant, or these flowers, they either self pollinate or cross pollinate, and they form and give you the desired fruits. Moving forward, in flowering plants, male and the female gamete are produced in the pollen sac and the embryo sac respectively. So pollen grain produced in the pollen sac, this is the male gamete. Alright, and in the embryo sac, you will find the female gamete. Hi Revati, how are you? Okay, so this is a structure. This we had discussed in the last class also. So this is the pollen grain. All right. This is the exine. This exine is made up of sporopollenin. This sporopollenin is one of the most resistant components, and it is resistant to heat, temperature, acids, and everything. This makes the pollen grain, it is like a support or a rigid outer covering for the pollen grain, which provides its resistance to almost everything, and it is also one of the most rigid components in nature. This forms the exine. Okay, now what you see, the aperture here is actually from where the pollen tube will generate. Alright, see, so this you see an aperture here, and from this a pollen tube has arisen. Okay, so the pollen tube have two male gametophyte. Here you have one and two, and one a sterile or a vegetative nucleus. So this is what a pollen grain and along with the pollen tube looks like. So this is a female gametophyte. In female gametophyte, this is the chelazal end and this is the micropolar end. So here you have three antipodal cells. Alright, you'll have two polar nuclei. You have a central cell. This is the egg, the female part, along with the synergids. Fine. Now these gametes are, see, what happens in case of animals is that generally the male gametophyte is motile. That means the male travels to the female gametophyte. But here what happens is both the male and the female are non-motile. By that I mean that they both are, they cannot be moved from one place to another. Therefore, there has to be a medium like wind or insect that has to take the pollen grain from one flower and deposit into the another. All right, so that is why pollination, the process is important, because both the gametes are non-motile. So again, this is the entire structure. You can see the stigma here. This is the pollen tube shown in red. Alright, so these are the antipodal cells which we observe. You have the egg, that is the female. Then you have the synergid cells. So basically, yes. So the pollination basically is a mechanism to achieve this objective, from which they can start reproducing. So the transfer of the pollen grains from the anther to the stigma is pollination. Alright, so now when this pollination happens between two different flowers, like from this anther, the male goes and pollinates this flower. This is the female part here, this is the male. So when the anther, from here the pollen grains will move and pollinate this flower. This is cross pollination, and maybe from here the male gametes will go and pollinate here in the female part. So this is what is cross pollination. It can be done in a single flower also, or in the same plant also. We will study about this in detail. So yes, moving forward, this is the complete slide is based on the NCERT and is very important. Most of the years, some objective questions are based from this. So what you have to remember is some plants such as viola, that is called as pansy, oxalis, and Commelina. You have to remember all these three names: Viola, Oxalis, Commelina. So what happens is they produce two types of flowers. Out of that, the first is chasmogamous flowers. So what happens here is these flowers are open. Alright, so the petals are broad and they are open. So all the parts inside, that is the style, pistil, ovary, anther, everything is exposed to the environment. Alright, so these flowers are open and are similar to most flowers of the other species with exposed anthers and stigma. So this is an example. See, here the petals are so broad and they are open. Alright, so the entire part, this is exposed to the environment. Similarly here, these also are protruding outwards, making it to open, and this part is exposed to the environment. So this is an example of chasmogamous flower and this is how it looks like. Now, what is the clistogamous flowers? Just the opposite. Clistogamous means the flowers in which the petals close themselves. Alright, so these flowers do not open at all. The best example for this is the green pea. So the garden pea or the green pea, you will always find closed flowers in this. Hello Bharat. Alright. So what happens is that in these flowers, the anther and the stigma lie close to each other. Yes, this is very obvious. See, what happens is since now these petals are closed, okay, it will look somewhat like this. Imagine these are the petals and they are closing itself. All right. So what happens is that, yes, so this is the anther and this is the stigma. They are pushed inside and they are forced to come close to each other. So when these anthers dehesize, by dehesize I mean that from the anther the pollen grains will burst open. Okay, when the pollen grain burst opens, these come in contact with the stigma. Since the male and the female, that is the anther and the stigma, is in very close contact, So as soon as the pollen grain ruptures the membrane of the anther, dehiscence is caused and immediately it doesn't have to travel so far. It will immediately come in contact with the stigma and pollination will take place. So thus, clistogamous flowers are invariably autogamous. By autogamous, I mean they will always self-pollinate. Alright, so what do I mean exactly by self-pollinate? So, when the pollen grains are transferred from the anther through to the stigma in the same flower. Alright, so this is self-pollination or autogamous. So, since these see, now these are closed. So this will never open. These flowers, the petals will never open. So it is never exposed to the outside world. So what happens is the mechanism of cross pollination is completely ruled out because the only place where it can travel is its own anther to stigma. So always there will in clistogamous flowers there will be asexual, autogamy or self pollination. So what happens is these clistogamous flowers produce a short seed set even in the absence of pollinators. So see, what happens is that since they are closed and not they cannot cross pollinate, the seed set, that means always the pollination will occur. Okay, there is 100% chance of pollination because what happens in during cross pollination we need to depend on insects, we need to depend on water or air or whatever. There is a requirement of medium from one flower to the another. Here both are in the same flower, so no medium is required, so there is a 100% chance of pollination. Depending upon the source of pollination. So pollination is of three types. All right. Autogamy, as I told you, when the pollen grains are transferred from the anther to the stigma of the same flower, it causes autogamy. The process of self-pollination or autogamy. See here. Now, this is an example of pea. See, you can see all the pea here. All right. So, what happens is this anther, okay, this here, from here, it will move here. So, the in the same flower, the anther to the stigma. All right. I hope this is clear. Moving to the next type. This is also geitonogamy. This is also an example of asexual as well as self-pollination. Fine. So what happens is, just a moment, yes. So what happens is that the pollination taking place here are between two flowers, but these two flowers are located in the same plant. So for this, obviously the plant has to be dioecious. All right. So what is the requirement? That a single plant should have a male as well as a female gamete. Then only they can assure pollination from two flowers, but from the same plant. So one plant only has to have both the type, that is the male and the female. So the best example of this is maize. It is not visible there. I'll write it down here. So the best example is maize. All right. So see, this is how. So this is one plant. All right. So in this figure, you can see all three types of pollination. So let us first look at a plant. What is happening here is that, see, from this flower, it is moving to this one. And both are of the same plant. So it is an example of geitonogamy since two flowers are involved. So I assume it is going from here to here. So this is contributing the male, that is the pollen will be transferred from here. All right, the pollen grains will be transferred from here to this is the female. All right, clear? So this is an example of geitonogamy. Now, here it is shown that, just a moment. All right, so here it is shown that it is coming to the, let me change the color so that it is more clear. So here in self-pollination, see, it is in the same flower again. All right, one single flower, when it happens from anther to stigma, is self-pollination. Now these are two different plants. Okay. Now what happens is pollinators will go from here to the other plant with the help of any of the insect or air or any medium. Now when two different plants are involved, then it is cross-pollination. I hope all three points are clear. Hello Akhil, hello Bharat. Thank you for responding. Moving forward, this is an example of maize. All right, maize is an best example for self pollination. Now coming to xenogamy. Xenogamy is nothing but it simply means, hello Nikita. It is completely okay if you address me as Janani. No need for ma'am. Okay, so xenogamy is basically cross-pollination. All right. So what happens here is that when the pollen grains are transferred from the anther to the stigma of another plant of the same species. All right. So it cannot be that one is say a tomato plant and another is a chili. Now this tomato plant cannot pollinate a chili plant. It needs to be of the same species. Then only cross pollination can occur. So cross pollination, what it is that it will lead to variation, since two different plants are involved. Okay, so the genetic content will change and it will lead to recombination. All right, so this is an advantage of, or this is an advantage of xenogamy. Agents for cross pollination. So what happens is, as I told you, that cross pollination cannot take place by itself, since both the female and the male gametes are non-motile. So we require a medium. All right. So these pollinating agents may be biotic elements, that means any animal or like insects, like honeybees or even snakes and such birds carry out pollination. All right. So abiotic factors can also be involved and are mostly involved. Those are wind and water are the most common abiotic factors. So majority of the plants use biotic agents, that to be precise, generally insects and birds are preferred. Okay, so this is an example of pollination by a bird and this is by a honeybee. Hello Nikita, hello Aditya. Agents of cross pollination. All right. So as I told you, abiotic and biotic factors are involved. Biotic factors involve bird, insect, and bat. And and abiotic factors are mainly involve wind and water. Now coming to the first abiotic agent, that is the anemophily flowers. So basically, by the word anemophily, I mean pollination by wind. So terrestrial flowers are mainly pollinated by wind. All right. So, so pollen grains are light and are non-sticky. When the pollination has to occur through wind, the pollen grains have to be light so that they can easily travel from in the air from one flower to the another. And they also have to be non-sticky. All right. They need to have a feathery stigma so that these light pollen grains come easily in contact with them. So, what happens is a numerous flowers are packed into inflorescence. By inflorescence, I mean uh, every flower blooms in its own fashion, or in simpler words, I can say it as it form clusters. All right. So, when these clusters are formed, they are termed as inflorescence. Okay, so when they are in clusters, it is very easy that these many pollen grains are deposited. Okay, it basically gives you a higher chance ratio. All right, so if not this, and there are many flowers, so even the pollen grain does not have to constrict itself to the to one flower itself. Okay, how efficient is wind pollination? I've already mentioned that it is mostly carried out by biotic factors. It prefers more because insects and that is insects and the birds. All right. So coming to this, the wind pollination you cannot always rely on because it will also depend on the direction of the wind. All right. So what happens is that if we have two plants here which needs to be pollinated by wind, all right. But if the direction of the wind is somewhat like this and not in which is not in either direction of the plants, so they will fail. And just imagine that there is a seashore or somewhat like this, and in this towards this side there is no similar species of the plant. So in such cases, wind pollination fail. So, and also it will again boils down to the pollen pistil interaction. So it is upon again, even if it is pollinated by the wind, it has to go to an identification procedure and it has to get accepted or rejected. All right. So what happens in a single ovule in each ovary? This is very much important in wind pollination. They generally have single ovule in each ovary. So hydrophily. So hydrophily, pollination by water. Just clearing one more thing. When we constrict ourselves to these abiotic factors, water and wind, between these two, by that I mean between water and wind, wind is the most preferred. That means more than water, wind has a more chance that it will cause pollination. All right. Now what? See, first of all, water pollination by water is very rare. All right, it is only constricted to about 30 genera or species, and these are mostly found in monocotyledons. Now let's see why. Because what happens is, even in the majority of aquatic plant, when they mature and when it's the time for fertilization, what it will do is it will come to the surface of the water. So once it comes to the surface of the water, it is the wind that will transfer the pollen grains and not the water. Hi, Cooking with Sumi. Thank you so much. Okay, all of you here need not address me as ma'am. You all can call me Janani. Moving forward, in majority of the aquatic plants, okay, as I told you, especially water hyacinth and water lily. What happens is that it will emerge above the level of the water and hence it will be pollinated by wind as discussed. Water hyacinth, see, it comes to the surface, and when it comes to the surface, it is the wind that will cause the pollination. And this is an example of water lily. Now coming to the types of hydrophily. Hydrophily is again divided into two types. There is some differences between the two. Okay, Bharat. So what happens is that epihydrophily, the first type, what happens is when the pollination that occurs on the surface is called as epihydrophily. The best example is Vallisneria. So what happens is that the female part will reach the surface of the water. Okay, so a female, it will develop a long stalk. Okay, see, this female will develop a long stalk like this. Okay, and this will reach the surface of the water. So this is the water, and this female part is above the surface of the water. So now what happens is that now the male parts, okay, the anthers are somewhat here. Now these, they, when this burst opens, the pollen grains are somewhat here, and they, with the help of water itself, okay, now like just imagine that there are either some small ripples or waves, and this water will cause them to come in contact with the female part. Yes, so it is carried passively by water currents. All right, so some of them eventually reach the female flowers and the stigma. So see, it is a chance process. Okay, here in cross pollination, be it any type, we cannot guarantee, or there is a very less chance that it will undergo pollination and it will be successful. But in case of self pollination, especially the autogamy one, it is 100% seed formation is assured. This is how epihydrophily happens. So this male has released its pollen grains, and these pollen grains come to the surface of the water. And these flowers, that is the seed. This female part has arisen up. All right. So this is the female, and from here, this will travel and reach the female part through water currents. Now, hypohydrophily. Let us see how it is different from the previous one. What happens is that you need to remember the examples. I stress on it again. Sea grasses such as Zoostera. Alright. What happens is that the female flowers remain submerged in water. Previously, what we saw was the female flowers rises above the water level. Here, they are submerged under the water. And what happens is that these pollen grains are released inside the water. This case, released inside the water is similar in the previous case also. Pollen grains in many such species are long, ribbon-like structure. Alright? And they are carried passively inside the water. Same. That means by the water currents, and some of these manage to reach the stigma. Okay? And they achieve pollination. And this type is called as hypohydrophily in Zoostera. See, now this, I don't know, it is quite faint, but these are the pollen grains. You see your long ribbon-like structures. Okay, now here the stigma is submerged under water itself, and these pollen manage to reach to the stigma. So what happens is that in most of the water pollinated species, uh, you see it is what to prevent it from water, pollen needs to have a covering so that it will prevent it from wetting. That is caused by this mucilaginous covering that will prevent it from wetting. All right, and another thing we need to remember is that in the previous class, I told you that one of the characteristics of the petals is that they are very colorful and they add to the beauty of the flower also, and also they attract insects. Even their smell, okay, the nectar, the smell, they attract insects. See, now when the flowers are submerged in the water, there is no chance of any insect or bird pollinating it. It is the water that, and or in some cases the wind that will play the role. For both these cases, they need not produce nectar, color, or any smell. So these flowers are generally not colorful and they do not produce nectar. Now, what are the characteristics of entophilous flowers? What happens is that when it is pollinated by animals, that is zoophily, it is called a entomophilous flower. All right. So here we saw an example of an insect pollinating it. Okay. Now what happens is, since now here an insect is involved, what happens is that these are large, colorful, fragrant. Now they need to have nectar because that is what is the attracting agent. Fine. So if the flowers are small, a number of flowers are clustered into an inflorescence. Inflorescence basically is the fashion in which or how the manner of these flowers are clustered into. So zoophilus is one of the following types based on the type of the animal involved in pollination. So basically in zoophilus, it basically refers to any animal. It can be an insect, it can be a bird, it can be a bat, snake, anything. So bees are the dominating biotic agent. So bees are the most common agents for pollination. Again, a very important objective question. Okay. Yes. So the first type of entomophily is that pollination by insects. See, as I said, bees are the most common one. Here is a snapshot of bees pollinating or taking up the nectar and causing pollination. Orthenophily. Orthenophily simply refers to ortheno. The term refers to birds. So any of the birds pollinating the flowers. So birds also will take up the nectar and the smell and also will get attracted to the colors. Then comes the chiroptero, chiropterophily. All right, what happens here? It is pollinated by bats. Recently, this is one of the most talked animal, bats. We all are very fed up of this animal, but it does help in pollination. Then coming to malacophyly. Malacophyly refers to the pollination by snails. So many animals are involved in pollination and they all help in some or the other way. So this is an example of pollination happening through a snail. Moving forward, therophily. Therophily means pollination by squirrels. Alright. Now, ophiophyly means pollination by snakes. This is an example of ophiophyly. So you need to remember all these terms, the names followed by what are the agents. The examples of the plants is not important in these cases. Only you need to remember is what type of pollination is associated with what type of zoophila or what type of animal. And the most common one is the honeybee. Let us see a few questions related to this. Pollination in between two flowers present on the same plant is called geitonogamy, xenogamy, autogamy, dichogamy. Okay. So see, xenogamy refers to cross pollination. Alright. So same plant is referred. So this cannot be the option. So geitonogamy now let me tell you why not autogamy, because it is mentioned two flowers and autogamy takes place in one flower itself. All right, so the only valid option here is geitonogamy, and dichogamy we have not discussed any such thing related, so it is again eliminated. In water hyacinth, pollination takes place by water, snail, insect, birds. Though the water hyacinth is basically a plant, water plant, what happens is that it comes to the surface. So as soon as it comes to the surface, the wind or the insect pollination, the chances are higher. The type of inflorescence most commonly found in anemophilous plants is compound spikes, head, umbel or corium. This basically is a question from the 11th standard syllabus from the morphology part. I'm sure everyone must be remembering this. This is caused by the compound spikes. Hi Amay, welcome. Thank you so much. Moving forward, pollination by squirrels is called. So, ophiophyly, therophily, malacophyly, or entomophily. Alright, so the answer is therophily. Ophiophyly is related to snakes. Let me write it down for you so that you can remember. Snakes. Sorry. Ophiophyly is related to squirrels. Alright. Malacophyly is related to snakes and entomophily is related to insects. So the correct answer is thyrophile. Outbreeding devices. Again, a very important term. What happens is that majority of the flowering plants produce hermaphrodite, that is a bisexual flower. That means in the same flowers there is an anther and an stigma. That means a male and a female gamete. So what happens is most of the plants try that they go under the process of sexual reproduction and they themselves try to prevent themselves from getting into the process of asexual one, though they are bisexual. Why so? Because what happens is inbreeding depression. All right. So what happens in inbreeding depression is that it is the nature's uh, trait or the nature's way. What happens is that if there is continuous asexual reproduction, the nature always selects the recessive traits. All right. So the recessive traits are promoted, and because the recessive traits are promoted, there is a high chance of inbreeding depression, and there is no variation. Okay, no mutation. They cannot develop themselves. They cannot adapt themselves. The genetic content simply flows, and all the damage of the DNA gets transferred from generation after generation. That is why the flowers try to prevent the autogamy or the asexual type. So continued self-pollination results in inbreeding depression. Now coming to dichogamy. What is it? Flowering plants have developed the adaptations. Okay, what I mean by adaptations is that what happens in adaptation is that, see, uh, once these plants try to prevent asexual reproduction, they need to have a method. So there are many methods by which they will discourage themselves from self-pollination and encourage cross-pollination. So this is dichogamy. So what happens is that in dichogamy, the pollen grain and the stigma, they do not mature in the same time. So when the pollen is ready, the stigma is not ready, so it cannot occur pollination. When the stigma is ready, the pollen is not ready. So again, pollination cannot happen. So this is this process in which both have both of the maturation time is not synchronized is dichogamy. Thank you, Utkarsh. Now moving forward, either the pollen is released before the stigma and becomes receptive, that is protandry. If the pollen is released before, it is protandry. The best example is sunflower. And if the stigma becomes receptive much before, okay, the release of the pollen, then it is protogyny again. That the example is datura or solanum. See, here you can observe protogandry. The first day flower, what happens here is anther is shedding pollen and but the stigma is closed. Okay, now the stigma is open, but the pollen has already shed. Now there is no more pollen to be shed. All right. Protogyny, see, what happens is the first day of flowering, the stigma is receptive, but the anther is not ready. And in the second day of flowering, the anther is dehiscing. That means the pollen grains are released, but the stigma is not accepting. So this will now lead a chance for cross pollination, which will cause variation. Coming to the next adaptation to prevent self pollination is when the anther and the stigma are placed at different position. Just imagine if the anther and the stigma are in the opposite direction. There is a no chance, because both are non-motile, there is no chance that they will come in contact. So that is in simple, that is in the term for that is hercogamy. So what this is mainly the best examples are hibiscus and gloriosa. All right. This is an example of hibiscus and this is for the gloriosa. Both prevent autogamy. Autogamy means self-pollination. Now coming to heterostyly. What happens here is the third method or the third device to prevent the inbreeding is at different heights. See, I'm repeating it again and again. Male and female are not non-motile. Okay, they cannot move from their places. Okay, so now in what happens is that the stigma and the anther is in different heights. So once there is difference in heights, it is very difficult for them to move from one height to the another. So what happens is, example, Olidanedia primula oxalis. All right. See here, you can see clearly the difference in heights. This and these are the anthers and this is the stigma. Because of the difference in height, it cannot move. All right. Now, self incompatibility. What happens here is that this is the fourth method. It will make itself insterile. Okay, it loses the fertility concept and it will completely become incompatible. Okay, to prevent the asexual or the self pollination. The best example is abutylon. All right. Now, in diclining, what happens is that this is a genetic mechanism which will prevent self-pollination from fertilizing the ovules, but it will inhibit the pollen germination or the pollen growth in the pistil. So what happens is that here it will make its female matured and developed and ready for reproduction. But the male part is not developed, so the concept of self pollination is ruled out. So what happens is it has for this female some it generally prefers cross pollination. So from some other matured anther from some different flowers, it will come here and help this matured female to get pollinated. This is what causes diclining. Another device to prevent self-pollination is the production of unisexual flowers. So, if in a flower only contains male gametes or only contains female gametes, again the concept of self-pollination is ruled out. And if both the male and the female are present in the same plant, such as castor and maize, because they are monoecious in nature, that is bisexual, it prevents autogamy but not geitonogamy. So, geitonogamy is the pollination of pollen grains from anther to the stigma for different flowers but of the same plant. If there is any doubt, please let me know in the chat section. In several species, such as papaya, the male and the female flowers are present on the different plant. Okay, these are dioecious in nature. So the male is in a different plant, the female is in different plant. So now what happens is this condition, dioecious condition, prevents both autogamy and geitonogamy. All right. Now moving to a very important concept of pollen pistil interaction. Please don't forget to like the video. It really motivates me, and if you learned a new concept or if you liked any part of the teaching, please do drop a like without fail. All right. So moving to the pollen pistil interaction. See, this is basically a type of identification. So what happens here is the filiform apparatus. Filiform apparatus is basically present the micropylar part of the synergids. That is, what is the micropylar? See, you remember the structure of the female gametophyte, the below, there are three synergid cells. Yes, that below that is the micropylar region. All right. So what happens is that this will promote the entry of the pollen tube. So generally in animals, what happens is that the female and the male, they will identify and they will accept. They will cause the exception or the rejection of the ovule and the sperm. But in case of flower, what happens is that male is present. Male, that means I mean the pollen grain. All right. But the female gametophyte does not decide whether it has to accept or it has to reject. Instead, it is the pistil. It is the pistil that will decide whether it will first identify. And after identifying, it will decide if it matches. That means if it accepts or it rejects. All right. Moving forward, it is a dynamic process followed by promotion or inhibition, that is acception or rejection of the pollen grain. Now, I want to tell you the first let me see. Yes. This is the stigma. You can see the pollen grains here. All right. The pollen tube also is visible, I'm sure. All right. So, yes, another thing is ovary should be visible. That is and micropylar region, ovule tunicles, these are all the secondary part. All right. So the first step is the identification. That is done. Okay. Now, if this step is approved, okay, only it has to first approve. If it is rejected, the following steps will not take place. All right. So when this is approved, it can move to the step 2. So what happens in step 2 is that this stigma will secrete a sugar plus water, like an enzyme, or let me call it a secretion. All right. Now what happens is that this sugar plus water secretion will enter into the pollen grain. Okay. Now this will enter into the pollen grain, and once this enter is or absorbed by the pollen grain, now this pollen grain will try to increase in size. So see, as I told that pollen grain is surrounded with sporopollen, which is very resistant. So it is not the sporopollen that will try to expand, but it is the inner part or the intine that will try to expand itself. All right. And this enzyme will prevent it. Okay, so enzyme prevents it. All right, I hope till the second step it is clear. So this change the color. So okay, fine. So the second step is the secretion from the stick to the pollen grain. All right, these steps have to be very clear. Okay, now what happens is that the third step, what will happen is the germination of the pollen tube. Now, see, now as this sugary substance, it it secretes, it is absorbed. Now what happens is that now this pollen will just try to expand, and now the germ pores, you remember the germ pores. Germ pores are the region where the sporopollenin is absent. So now the germ pores is the one that comes into action. So what happens is that from here, this will give rise to or give help the pollen tube to germinate. All right. Now the fourth step is penetration. Now pollen tube has developed. Now what the pollen tube has to go? It has to go and penetrate inside the stigma. Okay, so the penetration is like the pollen tube will go to the stigma, to the style, to the embryo sac. All right, this is the short process of the uh, penetration. Now what happens is, we remember that we had two types of cells in the pollen tube. One, let me change the color again. Okay, so the two types of cell present in the pollen tube, one was the tube cell, which which gives rise to the pollen grain, and the other is the generative cell. Fine. What happens is that these tube cell and these generative cell will try and come to the embryo sac. And once it starts entering the embryo sac, what happens is that it will undergo, this is the fifth process, it will undergo mitotic division. So once it tries to enter the embryo sac, what happens is that the germ cell which we saw will mitotically divide into two male gametophyte. Clear? I hope this process should be clear. So the first it will identify, then stigma secretes a sugar water secretion. It causes the pollen to expand. Then the pollen will start the germination of the pollen tube. This germination will happen through the germ pore. All right. Now this will start penetrating. Once the penetration start, what happens? It will undergo mitotic division. Fine. Now coming to the fertilization. Fusion of the male and the female gamete. Entry of the pollen tube into the ovule. This we have seen. It is three ways in which the pollen tube can enter into the ovule. Alright. So the first one is the porogamy. Porogamy is basically the enter of the pollen tube through the micropolar region. Clear? When the pollen tube enters this into through the micropolar region, that is from here. All right, this is termed as porogamy. Now moving to the second part, it is a chalazogamy. From the name itself, it will enter from the chalazal end. All right. Now the third type, mesogamy. Okay, in mesogamy, it will enter through the integuments. All right, this is the fashion how it will enter from the integuments. This structure is the integuments. Okay, so these are the three ways from which the pollen tube will enter. Very important. You need to remember the names and from the region where it is entering. I hope this is clear. Um, all right. So the example for porogamy is Otilia, then comes Casuaria and Cucurbita or coconut. All right. Moving forward. Entry of the pollen tube into the embryo sac. Irrespective of any fashion, be it porogamy, be it chalazogamy or mesogamy, it has to enter the embryo sac at the micropolar end. See, in micropolar end, in the porogamy, it is entering to the micropolar end itself from the start. In chalazogamy, it starts from the chalazal side, but eventually it will come towards the micropolar end only. And here also it will pass through the intubements, but finally it will enter through the micropolar end itself. All right. So the entry of the pollen tube in the embryo sac is under chemotropic guidance. By chemotropic guidance, I mean that the synergids have the filiform apparatus and they secrete some chemicals. These chemicals are termed as chemotropic guidance. All right. So see, this is how. This is the pollen grain. This is the pollen tube. This is the female gametophyte. All right. This is the micropolar region, which we can see here. These are the intubements. This is the egg. Okay, this is the diagram taken from NCERT itself. Entry of the pollen tube into the embryo sac. Again, the same thing which we have discussed. See, now what happens is, you can see there is a degenerating synergid cell here. There is the egg cell here. You see these are the filiform apparatus here. All right, the male gametophyte present here and the female, that is the infertile one present. So what happens is that this degenerative nucleus first will degenerate and see this it will fuse with the synergid nucleus that the pollen tube discharge will occur. All right. So this is basically the process how the pollen tube will enter into the embryo sac. Artificial hybridization. What I mean that artificial hybridization, it is mainly some processes that is performed to improve the program or the conditions of the crops. What happens here is that during the like in an agricultural field, what they make sure is that the plant is only the desired or the pollen grains having a very good genetic composition is or a very good quality of pollen grain causes the pollination and helps the reproduction. Okay, so this is why they have developed some artificial methods, like we force the better quality pollen to cause pollination, and we ourselves have invented some way to uh, prevent the other ones or the low quality ones. So the stigma, it is protected from the contamination of unwanted pollen. How it is done? See, this is the first process. So what is achieved by emasculation technique is that the female parent bears the bisexual flower. All right. So what happens is that the anther is removed from the female flower bud before the anther dehisces. Matlab, before the pollen grains burst open, the anther itself is removed. All right, using a pair of forceps. So this step is known as emasculation, the removal of anther itself. See, this is an example of emasculation using a forceps. The anther itself is removed. So that, see, this is in case of if it has bisexual flowers. So that now the one chance of autogamy is completely ruled out. Emasculate flowers have to be now covered with a bag of the suitable size. Generally, the bag is made up of butter paper. All right. Why is this done? So that contamination is prevented. So this step of covering the desired flower with the bag, this process is termed as bagging. See, this is how, see, this with the help of a butter paper, this process of bagging takes place. You can see a flower here. This is completely covered with the bag so that contamination is prevented. When the stigma of the bagged flower attain receptivity, mature pollen grains collected from anther of the male parent is dusted on the stigma and the flowers are rebagged. See, now we have the good quality pollen. So now whichever pollen we want, we dust them into the anther ourselves through artificial. Like just imagine like you take a paintbrush, dip it in the desired pollen and just brush it. Alright. And now rebag it. Again cover it with a bag so that we can prevent uh, further contamination, or it can even happen that if you leave it open now, some other pollen grain might come, the undesired ones, and start its process. So what happens is that the fruit is allowed to develop, and if the female parent produces unisexual flower, then there is no need of emasculation. Then unisexual means there is now only the female present, so there is no need for removal of the anther. So this shows the rebagging. Okay. Now coming to the last and the very important topic from the CBSE board exam point of view as well as the NEET point of view. Double fertilization. What happens in double fertilization is that now there are two male gametophytes which we saw. Okay, now one of them, one after entering the synergids, what happens is that these pollen tube will burst and release its two male gametophytes into the cytoplasm. So what happens is that one male gametophyte will move towards the egg cell and it will fuse with its, okay, and it will complete the syngamy, or the simple words, it is the fertilization. So there are two male gametes out of which one go and it will cause the syngamy with the egg cell, and it will form the diploid, that is the 2N, that is the zygote. So male gamete and egg will cause syngamy and zygote is formed, 2N. Now this important step, this is different and this occurs only in flowering plants. What happens is that now, see, I told you there are two male gametophytes. One has gone and fused with the egg. Now, what happens is that the other male will go and fuse with the secondary nucleus. All right? And this secondary nucleus will give rise to PEN. PEN. This is primary endosperm nucleus and it is triploid in nature. Very important point this is. It is triploid in nature and 3N ploidy it has, and it is primary endosperm nucleus. Now this fusion is called as triple fusion because three nucleus takes place in this fusion. So male gametophyte, two nucleus are already present, secondary nucleus 2N plus the N from the male gametophyte. Triple fusion results in a triploid condition. So the difference between syngamy and the PEN is that there the product is diploid, here the product is triploid. Alright, see, just observe this very carefully. Here you can see two male gametophytes. Alright, these are the two male gametophytes present here. Just follow the arrow. One will go and fertilize the egg cell here, and one will go and fertilize the secondary nucleus here and form a 3N. See, this is the 3N endosperm structure, and one will form the zygote, that is fertilization or syngamy. So see, one is 3N, one is 2N. So in total, 5N structures or 5N is the contribution in total for double fertilization. Okay. Male gamete plus the secondary nucleus causes the PEN structure, alright, which is triploid. Since these two type of fusion, the syngamy and the triple fusion, takes place in the embryo sac, the phenomena is termed as double fertilization. Along with this, you have to remember that it occurs in flowering plants only. Both these terms are very important. 5 nuclei are involved. As I said, 3N from the PEN and 2N from the syngamy. Okay, that's it. That is the end of the topic. Now we'll just solve a few MCQs from various topics which are also the previous year asked questions. By previous year, not I mean the just previous year, it is previously or frequently asked questions in the NEET. Progamy is entered of the pollen tube into the ovule through dash. We have studied three types: progamy, mesogamy, and chalazogamy. So progamy is the type of entry through the micropolar region or the micropyle. Entry of the pollen tube into the embryo sac is under the guidance of chemotrophic, chemotactic, phototrophic, thigmotrophic. The correct answer is under the chemicals. The word chemicals is related to chemotropic. Alright. The precautionary measures in artificial hybridization is or are emasculation only, bagging only, both emasculation and bagging, rebagging only. So the precautionary measure include both the emasculation and the bagging. Actually, rebagging is also a point, but the most relevant one here is both emasculation and bagging, since the next option states rebagging only, and that is not right. Double fertilization was discovered by Navashin in Lilium and Allium, Lilium and Fritillaria, Zemaze and Magnifera, Nigella and Fritillaria. I did not discuss this point. The answer to this is very important. It is Lilium and Fritillaria. You need to remember this point. All right. Thank you, Nikita and Akhil and all the others. I hope you liked the session. Please do not forget to like the video. And yes, do join tomorrow at 11:30. We will be solving more than 100 questions from this topic, that is sexual reproduction in flowering plants. And it will include the frequently asked and the most important questions from the NEET point of view. Do not miss tomorrow's objective session. Thank you so much.